Good morning, everybody. Welcome to the seminar, and welcome to the seminar. We are having a bit of difficulties. That's why we are a bit late, but I hope we can get going in a moment. I do have Samar's presentation here. I need to download, and here we can get going. Just give us a minute, please. Okay, I'm working on this. Okay. Samar, if you want to try sharing your screen, open up the microphone and tell me if you can share it. Oh, here it comes.
Shall I try to share my screen?
Well, I have your presentation. It's opening. It's pretty big, but it's opening now. All right. All right. I think we are good. I'm gonna show it from here. Okay, Samar?
Super.
Right,
thank you so much.
Now, hold on. Let me share my screen again, and we get going, okay?
Yeah.
All right. So thank you so much, guys, for attending this seminar. This is the last seminar of this season. I have the pleasure to introduce Samar Sheet, Dr. Samar Sheet, who works at the DSMZ, which is the Institute for Microorganisms and Cell Cultures at the Leibniz Institute for that center. She got her PhD in Germany, and she works together with Stefan Winter in viruses that affect cassava in Africa, particularly. Both Samar and Stefan have made significant contributions already towards food security and improving the living conditions of farmers that depend on cassava in Africa. So I want to thank you, Samar, for all your work that you have done in cassava, and thank you for accepting this invitation to talk to us, okay? So I'm gonna change the presentation now. Go ahead, you have the floor now. Hold on a second, let me do this. Does it show well?
Not yet.
So you may want to open your presentation in your screen and you talk and you tell me when to change slides, and we go that way if you cannot see my presentation. You can see the presentation, Thiago, or Brandon?
No, Carlos, I think you might be sharing the wrong screen. Try to turn off the...
All right, I'm sorry. I am sorry. How about now?
Yeah, this looks familiar.
Brandon?
Yes, it looks good.
All right.
So
ahead,
thank you.
Go ahead.
Thank you so much for this invitation and this chance to explain about our research and our experiment regarding finding resistance against cassava brown streak viruses, cassava mosaic virus, and cassava brown streak viruses. So I would like to start first, so we can go ahead. So where is cassava and where is cultivated? Actually, cassava is a woody shrub. It's come originally from South America, and it was later in the 18th century introduced to the Africa, where it became a main crop in Africa. And as you see in the map, it grows not only in South America, it grows in South America and in Africa, and it also in Asia. And in worldwide, it's considered food for 8,100,000 people. So the next. So since the introduction to Africa, actually, this crop has a main impact in cassava. Next. Because cassava, it's really important for the viruses and for the people there, because it's considered as a source of living for the farmer there, as you see in the pictures. And this because cassava can grow easily without any condition in poor soils, as you see here. So it's really easy to grow this in Africa, and it's also easy to intercrop this plant with other also crop like mango, maize, and coffee in Africa. It's also considered a source of food. So they use the tuber and also they make it flour, as you see. And also next, you can see that it's used as a tuber for feeding for the human, also for the animal. Next, please. So also here, as you see, the viruses is not only the plant is not only used for a tuber, as you see from this small girl there, they use it as a leaf as well for feeding. So next, since the introduction to Africa, this plant was faced a lot of viruses, and there are two major viruses that was facing this plant. The first one is cassava brown streak virus. It's an epomovirus. You see it in the upper panel, and it's from Potiviridae, epomovirus genus. And then you see it in the leaves on the upper panel as well. It has a chlorosis and leaf vein. The main impact of this disease is in the tuber, making this necrosis in the tuber, making this tuber inedible. However, the other disease is cassava mosaic viruses, which is down, as you see down. It's caused from Gemini viruses, Gemini viridae. It causes chlorosis and mosaic symptoms on the leaf. The impact on the tuber in the middle panel here on the left is making this tuber reduction in the tuber size, the white one. So this is the impact of these two viruses in the crop. This can make losses up to 90% of the crop. So next, we start with the cassava mosaic disease. Where's cassava mosaic disease grow? It grows wherever cassava is grown in Africa. It's an endemic. So wherever we have cassava in Africa, you could find this disease. And it's caused by several distinct virus species. It's up to 7 species in Africa, and they all have the same effect on cassava. And you see, they have the same impact on making small root. They make the same impact making stunted growth on this line, as you see in the middle pictures. The main vector for transmission of this disease is the whitefly Panacea tabaki, as you see on the right side. So this whitefly is very efficient because one whitefly is enough to transmit the disease and cause a disaster in the field in Africa. So the efficiency of the whitefly is really high. So the control option for this disease is finding resistance. Next, but before we start speaking about the resistance, let me introduce you about the response of plant to virus infection. So when a plant can get infected with viruses, we have two phases, can be infected or can be immune. If the plant gets infected, then we can face two phases. We can speak about the virus side, and this is how the virologists speak about the virus, or we can speak about plant virus, and this is how breeders speak about a response of plant. From the virus side, we can see if the plant gets infected, then we have susceptible, and this make the virus is replicate high in this plant. So we have a high virus load, and it can be resistant where we have a restriction in virus replication and invasion in this case. So if we look to the plant, then we have two cases. Sensitive, when we have less symptoms. So when we have a lot of symptoms, so the plant express a lot of symptoms, this is sensitive, or we have a tolerant case where the plant doesn't show symptoms or show less symptoms. And as you see here, so the resistant plant can be somehow in the range between tolerant and sensitive. So we could have a resistant plant that has a low virus titer, however, still have a symptoms expression, so that we have to be really careful when we define our term to speak about resistance in this case. So now, next to the cassava mosaic disease and resistant and what we know about the resistant. The resistant for cassava mosaic disease is broad spectrum resistant, and this was one of the Stefan research in the 90s, and as you see here in the middle, we we were unable also to identify resistant plants regarding the susceptible. And not only that the resistant plants were identified, and by all this breeding and all this research through the whole African and worldwide, they were able also to classify this resistance. So next, we could see that the resistance is not only resistant in one, so there are 3 types of resistance: cassava mosaic disease 1, CMD1, CMD2, and CMD3. CMD1 is a recovery resistant, as you see in the right panel. So the plant can show infected in the middle, yeah. So it shows symptoms. However, this plant can grow out of the infection, and as you see in the upper part, the plant doesn't express any symptoms. This CMD1 comes from process with Manhot Glasgowi, which is white type. CMD2, it's in the middle one, is the highly resistant plant, and it was identified in West African landrace, and it's considered the most stable resistant in all breeding projects. CMD3, the last one, extremely resistant, and it was a process between CMD1 and CMD2. And Tabomen, CMD2 is the most widely used in all breeding projects in Africa, and it's implemented to have a CMD in the breeding population. So now we want to characterize this resistance for CMD, and based on our research and our experience, we know that the cassava mosaic trichomoviruses causing the cassava mosaic disease can translocate through the vascular system of the plant when we infect the plant by body grafting. However, this plant cannot replicate the virus, so we don't have a virus titer. We can't, so the virus is not replicating, and also this plant doesn't show symptoms. And when we try to trace this virus in the tissue, and when we did a tissue section from the stem and from the leaf, we know that in the highly resistant plant, we were not able to find any viruses at all. And this virus localization is here, you see it as a red dots. So the red dots reflecting the virus presence. Not only that we were able to localize the virus, we also were able to specify where the virus is. So if you go next more, you could see that using specific fluorescence antibody, we were able to see where is exactly the virus, and in the case in our cassava mosaic virus, they are in the phloem cell. So based on this, and based on our resistance, we know that CMD is no more critical and no more threat to cassava cultivation in Africa. And this was one of the last pictures that we get in Nigeria. This is a new released Nigerian lines that has CMD. So CMD is most distributed in all Africa, and it's no more critic to African cultivation. So back to CBST. Cassava brown streak disease, what we know, it's occurred now actually in next, it's happening in Eastern, Central, and South Africa, as you see in the red dot where we have the outbreak. What we know also next, that it is caused by 2 distinct species. So next, it's caused by 2 distinct species, is cassava brown streak and Ugandan cassava brown streak. Normally, they have similar symptoms on the leaf, and they have the same impact on the tuber by making necrosis. And this is what we see here. And the role of Pimesia tapaci in transmission of this disease is still not that efficient, because regarding or comparing with the cassava mosaic disease, you need a lot of whitefly to transmit this disease. And what we know that we don't have a highly resistant plant in Africa. We see this is a field in Africa which was highly affected with CBST in the lower panel of the photo. So how we can manage the disease? We need first to look for the cassava brown streak viruses, to look to the biology of the cassava, to the viral response, the environment and ecology. We need to look for the entry, establishment, and spread. We need to look also to control, eradication, and phytosanitation, mitigation, and clean seeds. And at the end, we need virus resistance. So entry, when we come, this comes with the question about the cutting and the planting material that comes to the area that it's free. So this is how normally the disease started. Establishment also happens with the infected plant material. The spread can happen by 2 ways. Long distance by vegetative propagation. because cassava is not only, the virus is not only transmitted by whitefly, it's also transmitted vegetatively, because this plant can be propagated vegetatively, and spreading infected cutting between farmer and exchanging between farmer can also help to spread the disease. Localized spreads is happened by the whitefly, and it also comes seasonally, because when we have a rainy season, when you have a high whitefly pressure, then the disease is spread more. And when it's another season, that this affect also the spreading and how the viruses move. So when we control the entry, establishment, and spread, we come to control option in the next. So the control option comes from the phytosanitation, and in this here you see in this point, this is a point where Stefan, with the farmer there in Congo, were able to identify a place where it was infected with CVSV, and this is a clean island. And by communicating with the farmer and taking the right decision by eradicating this field, they were able to keep this area clean until now. The next is the mitigation, by communicating with the farmer and the stakeholder to use a seed system that's healthy, and also to use host-free zone, cropping cycle, and also to advise them with the best variety that we have until now. And the end, and what we needed now, is the virus resistance. So the source of resistance, screening protocol, and also how to use this resistant and breeding cycles. So on this point, we get our project, New Source of Cassava Brown Streak Virus Resistance, which was funded by Bill and Melinda Gates Foundation. In this project, we want to search for resistance against cassava brown streak viruses, and our search was in the South American germplasm, especially in Ciat, where they have the core collection of the cassava there. So we tried to identify resistance in the South American cassava germplasm, and also to validate our resistance by taking this resistance to the field and see the durability of the resistance under field conditions. And in later stage, by curiosity, we want to see why this plant is resistant. So to do resistant, we have we need a screening protocol. Next. And this, we need a tool, and we need to know how to deal with cassava, how to propagate cassava, which age is the best age for infecting material to get the efficient result. And this, in our case, we decided for 2 months, 3 months old cassava. Next was virus infection. And the virus infection in this case, it's naturally with the whitefly Pinacea tabaki, but we need to do it in our laboratory, and we don't trust the whitefly. So we can do it by different methods, as you see, by agro or bioholistic or injection or grafting. And we decided for the grafting next. And as you see in the pictures, the grafting was done by body grafting or side grafting. Body grafting, where you use a systemic infected bud and we put it on the cassava. And to increase the efficiency of our test, we do double bud grafting. Scion grafting or side grafting, where we take a scion, has 2 or 3 infected buds, and then we graft it on the plant. And in this case, you just assure that your plant has enough virus inoculum, and you are sure that your plant is infected. So after knowing which age we should use for the cassava, how we deal with cassava, and which infection method we should use, the next was to know which virus isolate we have to use. So I already told you that cassava brown streak disease caused by 2 viruses, cassava brown streak, Ugandan cassava brown streak, but not only 2 virus species, also it's a lot of virus isolate. So from our experience, we know that cassava brown streak Mozambique 83 is the most severe virus. As you see here, it has a necrosis and it kills the plant. Regarding the other one, the UCDF leukemia. So our interest is not Pentamiana. Our interest here is cassava, and as you see here in the pictures, that also on cassava, it has the first onset of the symptoms, and this was also regarding the high virus titer, as you see in the graphs there. So the cassava brown streak Mozambique is the most severe virus that has the earliest onset of the symptoms and also the higher virus titer. So as I know now that which virus to use, the next which method I have to use to detect my virus. So if I want to compare ELISA with RT-qPCR, I know that ELISA is good enough and sufficient to detect all the virus species for cassava. However, when you compare this with the RT-qPCR, you see the sensitivity of the RT-qPCR to detect the virus in the upper leaf, also when you have a low virus titer. And this is exactly what you want when you want to check for resistance, because you want to be sure that you have the minimum amount of the virus in your tissue. So our method was qPCR. The next was to check where I should sample, which sample I have to use for detecting the virus. So cassava is a big plant having a lot of leaf. We have a stem, we have tuber. So our first was to check different leaf area, as you see, and we know that the virus is, as you see in the graphs here, it's concentrated in the middle part of the plant. And also checking this regarding several months, we know that the middle part normally has the higher virus titer. So next, not only checking the leaves, but also we check the stem and we check the tuber. And as you see in the pictures here, the tuber has the highest virus load. So the tuber is very important when we want to check for virus resistance, because even if the plant show a little bit of virus titer in the leaf, it's not meaning that this plant is resistant, because the tuber can still replicate the virus, as you see here. Next. So also, if you check different here, when we check different plants, we check stem and root, you see that also the plant like Namikonga, where the leaf doesn't have a lot of virus, and the root and tuber are less, the stem is still important. So based on this, we decided to use a qPCR for virus detection and to use a leaf, stem, and tuber. So we received our core collection from CIAT. We did as a control African line, also to check as a control for us. And we used all the tools that I spoke about it, from the virus to the sampling, and it was 2-cycle confirmation and identification. So next, after we infect the plant, we could see that cassava responds differently to cassava brown streak infection. And if I speak about symptoms now, you could see that there's a plant that's highly sensitive, that it's died directly after the grafting. And this is 10 to 14 days after grafting. And we have in the middle where the plant show the typical symptoms of CBSD. However, in the other side, you see the highly virulent plant that didn't show symptoms at all. So this is from the symptom side, but we correlate the symptoms with the virus present. And here you see that we were able to identify 2 groups of resistance. One, it's the immune line, where you don't have virus at all, not in the leaf, not in the stem, not in the tuber. So it's a virus-free. So this is completely free. We don't have virus at all. And this also doesn't show symptoms at all. And we call this immune line. So other type of resistance in the middle. So we don't have virus in the leaf, not in the stem. However, we have a virus in the tuber. So we call this resistant root restricted. So this is really scientific evidence, and really was also found in another example like citrus spicreva virus, where the virus was also in the root, not in the shoot. So if you look from the breeding opinion of view, so this group is not important in this case, because what we want to breed for a healthy plant doesn't have symptoms at all. And in this case, this root restricted one is not good for breeding. So our candidate is the first group where we don't have viruses at all. So next was to also to screen for the other viruses. So as I said, cassava brown streak disease is caused by 2 viruses, cassava brown streak and Ugandan cassava brown streak. Next, so what we did, the candidate that we identified for the first one, which with the cassava brown streak, we screen against the UCBSV, the Ugandan cassava brown streak virus. And as you see here, that we could find some line that they are resistant for one virus, however, they are susceptible to the other. So we could see differential virus resistant response in this genotype. Next, we were lucky to find some lines that they were resistant for all. However, when we check for the EACMV, because this EACMV is the most also present in the field in Africa, we know that they were susceptible. So here, as you see, we brought this plant to the African sky. We put them under African sky because we want to test them under field condition. And we took them to different stations in Tanzania, Uganda, and DR Congo. And we see that our results in the lab were confirmed in the field because we could have the same result. They were the line that they were resistant for CBSV. They stayed resistant for CBSV. However, as we know, they were highly susceptible for EACMV, as you see in the middle pictures in the up. So this is a plant, the South American line, were highly susceptible for EACMV. Next. So what we try to do, we are trying to understand the mechanism of the virus resistance in the South American germplasm. For that, we try to follow the virus inside the plant by qPCR, by virus titer, and we propose a model of virus infection. So the whitefly comes to the upper plant, to upper young plant, and infect the virus. The virus moves cell to cell, with the cell to cell movement, and then moves down long distance through the vascular system, through the stem, in the external phloem, reach the root, which is important in cassava, and then the virus load increase with the virus replication, and then the virus move up again to infect the systemic, to make the systemic infection and invade the other leaves up. So now we know how the cassava brown streak move inside the plant, but also we want to trace this virus movement not only in susceptible plant, however, resistant plant. And by subjecting the section from stem, leaf, and tuber, as you see here, comparing the resistant with the susceptible, we were barely able to detect the virus in the resistant plant. And when we specify next the place of this virus in this tissue, we know that the virus is only occupying the phloem companion cell. So the virus reaches the phloem, and specifically the phloem companion cells, and then he's not able to unload to make infection in the parenchyma cells and replicating. So we know that the resistance of the South American line is based on restriction in virus replication and also in virus unloading. Next. So based on all this experiment and all our evidence, we know now that cassava brown streak is a root virus. This based on that the virus load is higher in the tuber root in the CBSV susceptible plant. It's also that we can find in the highly resistant plant some cases that even if you can't detect the virus in the leaf, you still can find a little bit in the root. And also based on our resistant group that we only find the viruses in the root restricted one. So next is how to use this resistance in breeding for Africa. How to use this cassava, the resistant from South American line, to breed for African country, and how to develop a high throughput virus screening protocol to enable us to screen for 2 diseases. This was our project for next in cassava. Next. So why we need actually the African cassava? And why African cassava is important? African cassava is important because all of them has the CMD resistant. So this is something that is good, so we don't need to have it again. And not only this, all the African cassava are end products because they come after a long breeding projects, and they all have a good trait that farmer likes. So like high yield, dry matter, and high quality. So the African line are in product. So what is missing in this African line is only the South American resistance against CBSV. So next, our hope for breeding was to use this South American line and cross it with the CMD from African cassava. And this was actually done by our partner in the NextGene cassava, the CIAT, TARI, and IITA. And we were lucky to receive the first population next and also to screen the first population. And what we see that F1, we were able to see the resistance. And this give us a hint that cassava brown stick resistance is a dominant trait. Next. And actually, to do this screening, we need a fast forward virus screening workflow. Depends on two elements, precision and speed. Precision based on we know what we are dealing with, we know our viruses and we know how to do it. And speed because we need to shorten the breeding cycle. And as you see here, we receive seeds, we germinate the seed, we propagate, and then we develop our workflow based on 2 cycles. Identification cycle, we screen first in the first phase against the CBSV. The negative one goes to screen for UCBSV and EICMV. And in this case, as you see, we don't screen for 2 viruses. However, we screen to 2 diseases, the CBSD and the CMD. So the negative one that goes out from the phase 2 can enter the confirmation cycle. And in this case, we can screen again in our greenhouse, or it goes to the field. And in this case, we confirm our observation in the field. Based on this, as you see from the time scale, we need around 9 months from seed to field, from seed to the candidate. And this really shortened the time of breeding cycle. In the conventional breeding cycle, you need around 5 years to reach a candidate. And this really fast forward virus screening workflow. Next. Now, this is how the population, now we receive a population from our partner and we are screening, and we were able to identify some candidates from population 1 and 2, as you see. And as we have the population, next was to bring this to the field. Next. How we could approach the field. And in this case, we need to approach the field by identifying an area. And here Stefan identified 2 areas. The first one is this Etri Island, where it's adaptation site and propagation site. So this is virus clean zone. So we could propagate our healthy material there. And the other area that's in the lower panel, this is the screening site. It's a hot spot where we have the 2 viruses there. So it's really high pressure from the virus. So next was to bring the plant from tissue culture and to take it to the people there. So we teach the people how to deal with this, from lab, from tissue culture to the hand of the people, only with small equipment, with plastic boxes and with small tents. We were able to put this plant under the hand of the people and to let this reach the field. Next. As you see here, this is one of our lower-resistant candidates that we brought from lab to the land. And this was passed through this process from the tissue culture to the harden, and then they were brought out of the tank to the open field. And also, as you see here, when we have a high pressure of whitefly, and even when we have these plants perform very well, and it's not only perform under the condition very well, but also it stay healthy and the resistance status is stable. So this one of our results from this. This is another, so this is another example. The KBH 2016 B521, it's a cassava plant which is resistant against CBSV. However, it's susceptible against UCBSV. So when we brought this to the hot zone in Congo, where we have a high pressure, and with taking a good practice as a measurement, you still can push the panoramic aside. And this was a story from us, from 3-year propagation, and also caring about this plant. 3 seasons were harvested, and the plant were virus-free, and everyone was happy. So I reach my conclusion now, and I would like to say that there is a broad spectrum immunity against cassava brown stick viruses in South American germplasm. This immunity in South American cassava is durable and maintained under high virus pressure in the field and performed very well. This mechanism of resistance in the South American germplasm is based on restriction of virus duplication and movement in the phloem. So we know exactly why this plant is resistant, but we know also that this plant are highly susceptible against cassava mosaic virus. So all the South American germplasm lacking resistance against cassava mosaic virus. Crossing between the CMD and CBSD resistant plant happened, and what we see now, we know that resistance against viruses causing CBSD and CMD actually is a fact. Next. So when we look for now the technical and how we can approach a final line, by knowing what we are doing, dealing with seed and having a target and having a workflow, we could learn how to do it and then deliver the resistance to the field, teach the people how to deal with this and how to follow on this, and at the end, you can see the result. And at the end, I would like to say, next, the secret of improved plant breeding, apart from scientific knowledge, is love. So if we love what we are doing and love what we are dealing with, we can push any pandemic aside, you can produce a healthy crop, and you can make everyone happy. So for that, I would like to thank you all, and I'm ready to answer any questions.
Thank you, Samar. Thank you. I think we navigated quite well the circumstances. Thank you for seminar. I like that quote at the end. I'm reading is love, and I like that. Please post your questions in the chat. Let me see. There's already one or two questions here. Okay. Does the breeding for resistance for the two viruses affect the palatability of the new breed? Does it change the taste change? That's the question.
So as we see now from the first population that there's no effect on the taste actually, but this still depends how we cross which is a parent and how this can be segregated later. But from the first population that we brought to Africa, we could say that there's no effect.
Okay. So Jules says, great work. Have you tried chemical control? I imagine for the vector, because probably for the virus is difficult.
I think for the white fly is not really sufficient. So I think you can't really control this by chemicals. So it's not that efficient.
White fly, chemical control of white fly is pretty difficult, isn't it?
Yes. And also, as I said, the white fly is not really the main cause for the CBSD to spread. So I think it's more than also to control other things like management and how the virus spread and how we exchange stem cutting and all kind of this stuff.
Okay, Jessica here is asking a question that I also thought about when you were talking is, have the genetic basis for this being studied on the molecular level, and do you think all the sources of resistance from Latin America are the same?
So comparing the immune line, yes, I think all are somehow the same. So these 5 are, I think, is the same. So now we are going through this, trying to highlight the marker for this. So trying to find the genes, the common genes between this that is responsible for resistance. And this is something that we are running with our partner Xiaofei in SIA, also from the next gene. So this is still under discovery, and we could have some hits on which chromosome could be the gene is there.
Okay. Well, that's great. Yeah, I think that will certainly enable even faster breeding of cassava varieties once you have markers. There's another question here. Peter Kuleko says that thanks so much for the excellent presentation. In the past, it has taken several years of field evaluation to confirm the resistance to CBSD. Are you confident that the short-term evaluations in the field will be sufficient for field resistance conclusions?
Yes, I will convince you that it's sufficient. So normally when we work under control condition, you know exactly which virus species you are working with, right? So now I know which virus I use, and I exactly know when I infected my plant. Yeah. So when you leave it an open field and you leave it for the white fly, or it's from 1 year before, then you can't control the time of the infection. And also you don't know what you are dealing with. So if we screen for resistance, knowing that I'm dealing with the most severe virus and infect my plant, then I know that if it's susceptible, in 1 month, it's out of my selection. But if I'm working also with a virus doesn't show symptoms directly and show me symptoms after 6 months, I could consider this as a resistant until it failed in the next round. That makes the selection is really late in the field, because the plant could be infected and didn't show symptoms until the next year, maybe, or until the end of the season. Yeah. So I hope that I convince you.
Jules is asking, what are the countries that you're piloting this resistance, testing this resistance?
So we have it in Tanzania and Pari and IATA. We have it in Uganda, in Nakri, and we have it in DR Congo in IITA, and also in the places that I mentioned in my presentation, this adaptation side and screening side in the Plan di Rosisi.
So I'm gonna sort of ask my question, not a question, but for you to comment. This is a disease that is spreading from Eastern Africa, and the risk is that reaches other regions. What are the efforts that are being done to preemptively breed for resistant to CBSD in other parts of Africa, particularly Nigeria, which is an extremely important, for which cassava is an extremely important crop?
Yeah. So I think in Nigeria, they will start soon as well with breeding for CBSD, because I think they have the most now, the most resistant line against CMD. This is the 5 Nigerian line that is, for example, released soon, that they have also high quality, and they had resistant for other things like mealybug and all other things. So I think this is really end product. And if they screen now, they start. I know that some of people are already started. So to cross this with a South American line, I think in this case you can got your golden plant that has everything in. So I think it started now to take care also that the places where you don't have a CBSD, to prepare that maybe in the future CBSD can reach. So that's already implemented in the breeding project.
Yeah, we know through what happened to us in terms of pandemic, what can happen to plants. And I also think, I know that also although in South America, these viruses are not present, there is a deliberate effort to use some of these sources of resistance more actively, so they are prepared, okay, in case these viruses are brought. We have a question here. What affects more cassava, CMD or CBSD? And are there effects in plant height and photosynthesis?
Yes, so CMD makes the plant stand and grows less. But the main effect of CMD is on the leaves, actually. So you are targeting the leaf more than the tuber. The tuber is only by reduction the size of the tuber. So if you compare the impact, I will say that CBSD, it has more impact because the people use more the tuber than the leaf. If you look to all African countries, and the impact of the CBSD by necrosis in the tuber, so this can cause 90% of the yield losses. So it's not, the CBSD doesn't have effect on the height of the plant, because this plant can show symptoms normally, and it still can grow really high and vigorous despite that it's infected. But the main impact goes in the tuber, and this can be masked because you could see the plant looks happy and grows vigorously and show a little bit symptoms. However, when you come to the harvesting time, you harvest and you see all that your tuber is killed by the necrosis, and then you lost your yield. So I will say that at the moment, CBSD, it's really the major disease that needs to be taken care of, because CMD, as I said before, it's no more critical anymore, because CMD resistance is now, it's all in all African countries are used.
Victor is asking, have you looked at the possibility of the resistance breaking down in the future?
So resistance breaking, so when I screen for resistance for this immune line that I showed you, I didn't screen only for 2 viruses, actually. I also screen about different isolates. So if I will say at the moment, if it will break, I could see it, but until now, nothing happened. So I would say that it's stable, but nobody knows. But for me now, I feel like it's stable, and putting this in the field with different virus isolates also, because you know that in Tanzania, the isolate is different from Uganda is different from Congo. We know that from the feedback, the CIS plan since 2019, it's in the field and it's stable, the resistance is stable. So 3 years in the field, I think I have to say it's quite good to say that this plant is holding resistance against the CBSD and against the CBSV viruses.
Okay. Well, Jules have asked us to share your email. He's probably gonna communicate with you more. Any other questions before we close? I have several. There's several comments thanking you and congratulating you for the work you have done, and it's certainly amazing. I'm gonna share my screen one more time, just to let me see if I can get out of here. Okay. Okay. I hope you guys can see my screen. Thank you so much, Samar, for this wonderful presentation to close this fall series of seminar. We'll be back next spring in March 2023 with another series of 6 seminars. I appreciate your attendance. If anybody has any suggestions about potential speakers or topics that you guys want us to address, please send it to me. We always appreciate that, okay? In fact, this series was built in the way it was built because of a couple comments, suggestions that were sent to me after the last series, okay? So we'll see you, hope to see you next spring, around March, is that when we start our next series. Thank you so much for the presentation, Samar. Thank you for the attendance of everybody. You guys have a great rest of the week. I won't see you through the seminar until next year, so have happy holidays and best success in what you do in your work. Thanks, Samar, and give a big hug to Stefan from my side, okay?
Thank you so much.
Okay, bye guys. Thank you, Samar.
Bye.
Bye.
Thank you. Bye-bye.